Thank you, Charlie. And just to confirm that everyone can see my presentation on the screen.
Yes,
Okay,
yes, everything
wonderful.
okay.
All right. Well, thank you very much for inviting us to share some data with you this afternoon. We're quite pleased to be here. Charlie did an excellent job talking about my lab's interests in the acute effects of exercise on immunometabolism. And so I'm going to focus my portion of the talk on more of the effects that we think we may see with exercise training or cardiorespiratory fitness on immunometabolism. And then given some of the results that we think we might see, discuss some potential clinical implications of these data. So exercise training, particularly aerobic or endurance exercise training, has many recognized health benefits. For various bodily systems, including the cardiovascular system, skeletal muscle, the brain, as well as the immune system. And many of these benefits of exercise can be attributed to an increase in the quantity and the function of the mitochondria within the cells, which therefore increases the cell's capacity for energy production and supports vital cell processes. Cardiorespiratory fitness also generally increases with exercise, and so can be used as a way to gauge an individual's exercise habits. Within the immune system, exercise training and having greater levels of cardiorespiratory fitness are associated with improved immune function, as exemplified by both decreased rates of problematic immune activation, so for example, chronic low-grade inflammation, decreased frequency of senescent cells, and improved immune function, such as resulting in lower risk of infection, enhanced cell proliferation, and greater vaccine responses. The metabolic adaptations of the body with exercise and the changes that occur to the immune system are particularly intriguing, given the link between immune cell function and metabolism. And as Charlie has already alluded to, very broadly speaking, quiescent cells, for example, memory T cells or regulatory cells, largely rely on mitochondrial oxidation for their energy needs, whereas activated effector immune cells increase their use of glycolysis, perhaps both for the rapid energy production and also as the metabolic byproducts support the effector functions. However, it's not really an either or. Mitochondrial respiration is still needed to support these cells. And so there's some intriguing data that has occurred from some in vitro studies where manipulation of mitochondria, for example, within T cells, really highlights the importance of the mitochondria for cell function. So boosting mitochondria in vitro can improve some measures of T cell function. Removing mitochondria removes their ability to function. And so this really highlights the fact that increasing leukocyte energy production capability can be a way to increase cellular immune cell function. And so in vitro experiments are great, but is there a way we can do this in vivo? And so we wondered if exercise training could be one such means of enhancing energy production and thus improving immune function. And so to begin to answer this question, we conducted a scoping review of the literature. And as we suspected, we are not the first group to wonder if exercise alters the metabolism of immune cells. We found 41 original reports to include in this review. These 41 studies included those that measured metabolic regulation, metabolic function, and leukocyte function following either a single bout of exercise or after exercise training. And I'll note that in most cases, this exercise involved an aerobic component rather than resistance training components of exercise. The participants in the studies included athletes, healthy active adults, and people who were inactive or with disease. So a bit of a busy figure shown on the slide at the moment. What we're seeing here is the number of studies that fall within each domain. So the number of studies that looked at, for example, metabolic regulation that occurs during exercise training amongst athletes or amongst healthy active individuals, so on and so forth. And so very broadly, the results suggest that acute exercise can influence the regulation and function of leukocyte metabolism. And this has some similarities to what has been previously documented in skeletal muscle. And Charlie has already discussed some of these results, as well as how our team is working to further our understanding of the effects of acute exercise. There's also data to evidence cardiorespiratory fitness, which likely reflects the results of exercise training, can also alter cellular metabolic regulation and function. So looking over at the right-hand side of the screen, not quite as many references, not quite as many of studies fall into these categories, but to highlight results from these few studies in this area, generally we see that people with greater activity levels or people with greater levels of cardiorespiratory fitness across different age groups do show evidence of greater lymphocyte mitochondrial mass or measures of mitochondrial activity compared to people who are less active or who are less fit. We also noted several gaps in this literature. In particular, there is few studies which examined the effects of physical fitness on both leukocyte metabolism and leukocyte function within the same study. And the potential influence of cell subsets on these measures were not taken into account. Many studies have also only included men, and other cellular metabolic processes outside of the mitochondria, for example, glycolysis, have not been as frequently considered. Another PhD student of mine, Justin Crane, is seeking to address these particular gaps as he is aiming to better define the relationship between cardiorespiratory fitness and CD8 T cell metabolism and function. So to answer this question, if cardiorespiratory fitness relates to T cell metabolism and function, we've invited healthy men and women who are between the ages of 20 and 40 to participate in this study. We assessed VO2 max via a maximal cycle ergometer ramp test. And in our sample that we have collected so far, with 43 people, these VO2 peak scores ranged from 23 to 58 milliliters per kilogram per minute. And we didn't see differences in our sample in VO2 peak values between men and women. Before they underwent the maximal exercise test, they also donated a resting blood sample. And from this resting blood sample, we isolated PBMCs and we analyzed these by flow cytometry. As noted earlier by Charlie, leukocyte metabolism differs between quiescent and effector cells. And so with flow cytometry, we identified different subsets of the CD8 T cells, including those that we expect to have less effector functions. So our naive cells or essential memory cells, and those with a greater amount of effector activity or effector potential activity are effector memory cells and our EMRA cells or CD45RA re-expressing effector memory cells. Within each of these subsets, we measured mitochondrial volume and function based on mitotracker dyes that are specifically staining mitochondria. We use the technique CNIT, which, as Charlie described, yields several variables related to cellular metabolism based on pyrimyosin uptake following the addition of various inhibitors. And we measured the ability of cells to respond to stimulation by producing interferon gamma, which is one of the primary effector cytokines that CD8 T cells produce. So I'm going to share some preliminary results. As these are preliminary, I ask you to please do not share. Our simple linear regression models show that VO2 peak, which was our measure of cardiorespiratory fitness, does significantly predict the oxidative capacity of each of the CD8 T cell subsets that we examined. So those with greater CRF, cardiorespiratory fitness, demonstrate greater oxidative capacity within each subset. So shown on the screen now are the results of the simple linear regression. We also conducted multiple regression to control for participant sex and percent body fat, and the VO2 did remain a significant predictor in those models. We also found that VO2 peak predicted the ability of some, but not all, of the CD8 T cell subsets to express interferon gamma in response to stimulation. So this was significant, except for the central memory and our effector memory cells. Potential explanation reasons so interferon gamma production is just one way in which CD8 T cells respond to stimulation. So it could be that these cells are responding in a manner related to cardiorespiratory fitness, and we just haven't captured in our data how they are responding to stimulation. So far, our results indicate that in the healthy young adults we've recruited, cardiorespiratory fitness is associated with CD8 T cell oxidative capacity as well as cell function. And so if these results can be translated to people with disease, it could be that exercise training which results in increased cardiorespiratory fitness, can be a way to enhance T cell function via enhances in T cell metabolism. So going back to the scoping review, there has been some research conducted, which has asked if exercise training alters leukocyte metabolism among individuals with disease. So in particular, we found 4 studies that examined leukocyte metabolism in people with disease before and after exercise training. And again, results generally suggest that there are improvements in cellular respiration, especially when that training resulted in increased cardiorespiratory fitness. There are also apparent gaps in the literature, as noted before. So for example, only one study also examined in the same publication, cellular function as well as cellular metabolism. And once again, the particular effects of different subsets of immune cells haven't yet been considered. Another result that stood out to me was this particular finding here from Hosny et al., which found that greater mitochondrial respiration is indicated by OCR, or oxygen consumption rate, which increased after training, correlated with improvements in fatigue in this patient population. Others have also found relationships between cellular energy production, particularly that which occurs via mitochondria, and fatigue amongst a variety of different patient populations, including cancer patients. Many cancer patients suffer from a particular form of fatigue called cancer-related fatigue. A few years ago, one of my collaborators, Tamara Lacourt, published some intriguing data which link PBMC metabolism and cancer-related fatigue in cancer survivors during and after chemotherapy. In this study, women scheduled for neoadjuvant chemotherapy were followed before, during, and 6 months after their treatment, and they were asked at these time points indicated here about their fatigue levels, and they donated blood samples. In these blood samples, leukocyte metabolism is examined. Oxidative phosphorylation spare capacity, which is a reflection of the ability of mitochondria to respond to increases in energy demand, decreased during chemotherapy and are improving a bit during the recovery period post chemotherapy, but as you can see, don't quite fully recover, at least in the time points examined here. And importantly, this lower spare capacity was associated with greater fatigue after treatment. So these results also suggest, though, that if there's some intervention or some strategy to improving spare capacity, to improving cellular mitochondrial respiration, then perhaps we could help alleviate fatigue, cancer-related fatigue in these patients. But clearly, this requires further study. And so we plan to do just that. Tamara Lacourt, Maria Swartz, some other collaborators of mine, we're planning an exercise intervention that we hope will alleviate fatigue and improve leukocyte metabolism. In this study, we're particularly interested in young adult brain tumor survivors who have cancer-related fatigue, as interventions specifically for this population are lacking, and cancer-related fatigue is a major issue for this group. We plan to quantify our intervention's effect on fatigue, on physical activity, physical function, and on our measures of leukocyte metabolism. So oxidative phosphorylation is one example. We'll look at a few others as well. And we'll be measuring immune function, so inflammation and some other ways of looking at immune function. This study has just recently been funded, and so we're in the very early stages of getting this project up and off the ground. So hopefully in the next couple of years, we'll have some exciting results that we can share in future presentations. To summarize, exercise training changes cellular metabolism in many different tissue types. And given the fact that leukocyte function and metabolism are really tightly linked, and that exercise training changes immune cell function, we and many others are particularly interested in understanding more about how exercise training can alter leukocyte metabolism and how that relates to changes then in immune cell function. In particular, a lot of groups have noted, including our own, that those with greater amounts of exercise training resulting in greater cardiorespiratory fitness appear to have enhanced mitochondrial metabolism. And so this leads us to potential clinical implications, in particular, perhaps those who are suffering from fatigue. But again, a lot more work to be done in this area. So I'd like to acknowledge the hard work of my current and former students. So you all have met Charlie already, who's done a lot of the groundwork for this project. I also presented results that were led by Justin Crane. And in particular, like to also highlight the efforts of Rebecca Maddox on this project, and of course, acknowledge and thank funders for this work.